This is the last video for electrophysiology, and this session will be discussed on properties of action potential. So these are the properties of action potential: all or non response, stereotypical size and shape, refractory periods, and propagations and conductions. All or non-law. Action potential exhibits all or non-law. Action potential either occur or doesn't occur at all. If an excitable cell is depolarized to threshold, then the occurrence of action potential is inevitable. If the membrane is not depolarized to threshold, no action potential can occur. After the threshold level is achieved, the amount of sodium influx becomes independent of stimulus factor. So this property is known as all or none law. Next is stereotypical size and shape. In an specific excitable tissue, the magnitude, duration, and then shape of action potential does not change. Frequency of action potential does change, which depends on intensity of stimulus, but the amplitude of action potential remains the same. This is known as stereotypical size and shape. So here, one threshold stimulus, there's a one action potential is generated, while when there is an increase in stimulus intensity or supra-threshold stimulus is applied, there is an increase in frequency of action potential, but the amplitude of action potential remains the same. Another properties is refractory periods. It is the period during which new action potential cannot be initiated by normal events in a region that have just undergone an action potential. Hence, two action potential cannot overlap or fuse. And here in this picture, the shaded portion is the refractory periods, and it is two types. One is absolute refractory period and another is relative refractory period. Absolute refractory period, the period from the threshold till the 1/3 of repolarization. This is known as absolute refractory period, means during this period another action potential cannot be generated by any strength of stimulus. While relative refractory period from 1/3 of repolarization till the resting membrane potential, during it is the period, short period after absolute refractory period, when the cell is refractory to ordinary threshold stimulus but responds to stronger stimulus, means the cell is responsive to suprathreshold stimulus. These refractory periods is mainly due to the changing status of voltage gated sodium channel during and after action potential. All the sodium channel does not achieve activated, inactivated, or resting state exactly at the same time. And because of this refractory period, in a neuron there is a one-way propagation of action potential from initial segment of the axon hillock to the terminal side of the axon. The last one is the propagation and conduction of action potential. The propagation of action potential down a nerve or a muscle fiber occurs by spread of local currents from active regions to adjacent inactive regions. Means the nature of propagation is electrotronic spread. Conduction velocity of action potential in a nerve fiber depends on nerve fiber diameter and myelination. Greater the diameter of the nerve fiber, greater the conduction velocity, and myelinated nerve fiber shows the conduction velocity faster than that of the non-myelinated nerve fiber or unmyelinated nerve fiber. In the unmyelinated nerve fiber, the conduction of action potential is point-to-point conduction, also known as contiguous conduction, and in myelinated nerve fiber, it's a faster node-to-node or saltatory conduction. Let's see in a diagram. So this figure shows the propagation and conduction of action potential in unmyelinated nerve fiber, and the second one is myelinated nerve fiber. Conduction of action potential in unmyelinated nerve fiber is contiguous, means action potential propagate in every patch of the membrane, and it's relatively slow propagation. While on the other side, action potential is initiated in the initial segment of the Eisenhallock, and then it jumps to the from one node of Ranvier to the next node of Ranvier to the next node of Ranvier, so that there is a jumping conduction, also known as saltatory conduction, and it's relatively 50 times faster than that of the unmyelinated nerve fiber. And in this active nodes at the peak of action potential, so it jumps to the next adjacent inactive area, and there is a genesis of action potential. In the previous region, it remains refractory to the ordinary stimulus. So this refractory period ensures one-way propagation of action potential from initial segment to the terminal side of the axon. The saltatory conduction provides the two advantages. The first one is, for a given diameter of a nerve fiber, it gives a higher conduction velocity. For example, 1 micron diameter myelinated nerve fiber conduction velocity is about 6 meter per second, while for an unmyelinated nerve fiber, same 1 micron diameter, the conduction velocity is about 2 meter per second. The second one is, this saltatory conduction conserves the energy. Since action potential jumps from one node to the another nodes, ionic changes also occurs only at the nodes of Ranvier. And this ionic changes during action potential is corrected by sodium potassium ATPase pump, so that the activity of this sodium potassium ATPase pump to restore normal ionic concentration is also reduced because of this saltatory conduction. So these are the two advantages of this saltatory conduction. So with this we come to an end of all content on electrophysiology, starting with the membrane potential and resting membrane potential, next action potential and ionic basis of action potential, and the last video that we have discussed today is about properties of action potential. So hope for the best. Thank you all.